Oh, hi everyone. So this is the last lecture in techniques in microbiology. So this lecture will cover bacterial count, so which means we will learn the techniques on how to count number of bacteria in a sample. And this lecture is quite relevant to practical too. Okay, bacterial counts can be performed either using a viable count or a total count. So viable count means we are counting only the leaf cells. And then the viable count including pour plate, spread plate, membrane filter or filtration, droplet method or also known as a mass in Misra, and most probable number in the short form is MPN. And just take note that you will do pour plate and spread plate in your practical too. And when it comes to the total count, we are counting whichever that we can observe under the microscope, which means we are counting leaf bacteria as well as the dead bacteria. And usually that is performed under the help of counting chamber, or also sometimes known as a haemocytometer. So one of the common practice in viable count is serial dilution. So objective of serial dilution is we dilute the concentration or density of the bacteria so that we can count in the plate. So let's look at a pop plate. So usually after we do the dilution, so we transfer a particular amount of the inoculum into the empty petri dish. Okay, and after that we add the melted nutrient agar into the petri dish, and then we swill to mix the inoculum as well as the melted nutrient agar. And after you mix it, we leave it for like 10 to 15 minutes for the agar to solidify, and after solidify we put it in the incubator. And after 18 to 24 hours we count the colonies, and usually the colonies grow in and on the solidified medium. And on the other hand, the spread plate method, we transfer a limited amount of inoculum into the plate that's already containing a solid medium, which is the agar. Now after that, we're using a spreader, and also some people calling as a hockey stick, to spread the inoculum evenly on the surface of the agar. And after that, we incubate for 18 to 24 hours, and usually the colonies grow only on the surface of the media. So now let's see how we can analyze the results. So the unit to measure the number of bacteria is number of bacteria per mil, or colony forming unit per mil, or the short form is CFU per mil. And this is the formula. So average plate count times with reciprocal of the dilution, or can be dilution factor, times 1 divided by the plating volume. So look at this form, look at this one now. So let's say the average plate count is 97. So times with the dilution factor 10 to the power of 4, and then 1 divided by the plating volume is 1. So and then you get like a 9.7 times 10 to the power of 5 organisms per mil, or you can also put it as a CFU per mil. Both are correct. Okay, there are a few factors will affect the number of colonies in spread plate and pour plate. First of all is the inoculum size, and in particular is the overcrowding of colonies. So when it comes to the spread plate and pour plate, so we're always taking into consideration 3 to 300 colonies. If it's more than 300, we always put TN TC, which means too numerous to count. So when the colony is more than 300, so there must be error happen when we are counting. And the second thing is the suitability in the culture medium, and this is because if we need to provide an optimal medium for the bacteria to grow, or else the bacteria won't grow or they will grow slower. And the following one is the incubation condition, and this is similar to the media. So we need to provide a condition which is optimal for the bacteria to grow, or else they won't grow or they will grow slower. And last and the most important is the rate of growth of the organism. As I mentioned before, E. coli is a fast grower. So generally, when you're doing a spread plate or pour plate for E. coli, you should able to get the results in 18 to 24 hours. But then on the other hand, let's say you're using a slow grower, so you may need to wait for like a month to see all the colonies. Okay, the following method is filtration. So filtration is hardly used in the clinical environment, but then it's quite common in the aquatic microbiology. So usually what happens here is that the sample either can be a fresh water or a salt water will be filtered using a membrane or millipore filter. And a commonly used filter is the cellulose acetate with a pore size of 0.22 micrometer or 0.45 micrometer. So what happens here is, so when you filter the samples, the liquid samples, so the bacteria will be trapped on the filter. And then when you put the bacterial trapped filter on the culture medium, so the bacteria will start to grow. And then after the incubation, you should be able to see the colonies, and then we count the colonies, and then we use the formula, CFU per mil formula, we count the number of bacteria. So this is how the filter looks like. Okay, and this is the apparatus we commonly use. So where you put the filter on top, and then you put the whole thing on top here, and then we filter through. Okay, this is how the apparatus looks like. So we put the filter inside the filter funnel, and then we pour the sample here, and then the whole setup will be connected to a vacuum pump. So when you turn on the vacuum pump, so we'll create a vacuum here, which eventually will suck the liquid in. So when the liquid is flowing through the filter, whichever the larger than the pore size will be trapped here, so which is the bacteria in the case. And after that, the filter, we put it on the agar media and then incubate for 18 to 24 hours. And after the 18 to 24 hours incubation, you should get something like this. So all these red dots are the bacteria, are the bacterial colonies. Okay, the following method is drop plate method. And drop plate method also known as a Miles and Misra. So this drop plate method is quite similar to spread plate, but then instead of using a hockey stick, the serially diluted suspension will be dropped onto the surface of the agar. And after that, the agar will be allowed to dry at the room temperature and then put it in the incubation. And one of the key thing in drop plate method is the amount of the liquid that you're dropping onto the plate. So usually the maximum liquid we keep it into 20 microliter. And in order to avoid overcrowding, only colonies between 2 to 20 colonies were taken into consideration for the CFU per mil calculation. Okay, this diagram summarizes the drop plate method. So as usual, first we serially dilute the sample, and then instead of spreading using a hockey stick, so we put like a tiny droplets into it. And then as I mentioned earlier, the volume of the inoculum must be standardized, and the maximum volume usually 20 microliter. And after incubation, you should be able to see something like this. So then this spherical thingy, the each colonies will be counted and CFU per mil and then calculated. Okay, the following method is most probable number, or simplified is MPN. So usually MPN involves inoculation of multiple tubes with replicate of samples of dilution. So patterns of tubes that shows growth, we put positive, and those that do not grow, we put negative, compared with a statistical table to calculate the MPN of bacteria in the original sample. And also remember, like sometimes the statistical table also known as a conversion table. Okay, this diagram shows how an MPM can be performed. So this is your undiluted sample, and then as usual we dilute them like 1:10, and then further dilute 1:100. And after that, 1 ml of the sample will be transferred into another test tube which containing a phenol rate. So the function of phenol rate here is a dye, it's an indicator, tells you whether the bacteria grows or not. If the bacteria doesn't grow, the phenol rate will retain rate. If the bacteria grows, the rate will turn into yellow. Okay, so we transfer the one diluted as well. Okay, and after that we incubate for like 18 to 24 hours, and after that we see whether there's any color change or not. So let's look at this results. So if there's a color change from red to yellow, it indicates positive. If it's a remain red, it indicates negative. So this set, so we got like a 4 positive tubes, and this one we got like a 2 positive tubes, and this one we got like a 1 positive tubes, and then literally you generate a number. So which number is 421, and then this number we're going to be using into the statistical table. Okay, this is how the most probable number MPN number table looks like, and some people also call it as a conversion table. So in the previous slide we obtained a number 421, so which is equivalent to 26 most probable number index per 100 ml. Okay, the final one is the total count. So total count, usually we are counting whichever that we see under microscope. So usually we count the dead cell as well as the live cells, and the apparatus or the glassware we're going to use for the total count is the counting chamber, and some people also call this as a haemocytometer. And then usually the haemocytometer will look something like this. So it's usually made of glass, and when you look under microscope this area, so you're going to obtain something like this. So you'll see a lot of squares. So usually this area the important areas are the 1, 2, 3, 4. So these are the areas going to be taken into consideration for the bacterial cell count. Okay, this slide shows how we can do a total cell count using a counting chamber or haemocytometer. So first we take the haemocytometer and a glass cover slip and clean it with 70% ethanol and make sure they're fully dry after that. After that we place the cover slip over the counting chamber and pipette 10 microliters of cells or bacteria into the haemocytometer. And one of the common practice is that we also add dye in the bacterial or cell suspension in order to get a better contrast when viewed under microscope. After that, we put the whole haemocytometer under the microscope and view under 100 times magnification in oil immersion. And under the microscope, we should be able to see something like boxes like this. And then we count the number of bacteria in 1, 2, 3, and 4 squares here. And then using this formula, we calculate the cells per mil. So usually like square 1, square 2, square 3 and square 4 divide by number of square is 1, 2, 3, 4. So 4 squares times to the power of 4 and then you will get the cells per mil. And usually haemocytometer is the fastest way, but then the only problem is that you need to buy the haemocytometer, which is sometimes not sometimes, most of the time is relatively expensive. And then also it is made of glass and it is quite fragile. Okay, we are done with viable and total cell count. So as for now, remember that viable count is valid if you are concerning about only living and metabolically active organism, which is very crucial for clinical samples and food samples. On the other hand, total count is in order to know the total number of cells in a particular sample, so which is including dead and live bacteria, and is quite relevant when you want to study the microbial ecology. So we are done with techniques in microbiology. So if you have any question, please post in the e-learning. And again, my apologies if the lecture is not clear, but then don't hesitate to ask questions so I know which part is not clear so we can discuss further when it comes to the synchronized face-to-face session.